everyone. We're going to go over chapter 14 and 15 today. I know it's two chapters, but I combine both of those chapters from the book into one just bigger chapter. This chapter doesn't really have a good flow of material, meaning like I can't do section one, section two, section three, section four. There's just so much randomness here because biotechnology and all of the different fields of biotechnology can be very separate and sometimes can cross over. But this is kind of a weirder chapter, so just kind of stick with me here. I will try to upload some smaller videos because in class I'll talk about something and then I'll show that process in a or short clip on like YouTube because there's just so much information here. But again, I only really want you to know the basics of biotechnology. All right, so first, genome. When we talk about genome, we're talking about the just the complete set of genetic material for an organism. The human genome is around 3.2 billion base pairs long. So this is talking about every nucleotide from the first nucleotide of your first chromosome to the last nucleotide of your last chromosome. So you have 46 chromosomes, 23 pairs. It's talking about every single nucleotide. Now, that's going to be different, by the way. This is going to be different. That 3.2 billion is only specific to humans. And each human is going to have a different number, but they're relatively all going to be pretty close, around 3.2 billion. Now, the Human Genome Project was actually started to figure out this sequence was actually started back in the 1990s. It took over 10 years to complete the first sequence or the first person. If I remember, I think it was a French person they chose. But it took multi-billion dollar corporations, many nations, 10 years, and it cost about $3 billion. So it cost a lot of money for this first genome to be completed. And since then, it's drastically decreased. And you can see this is even back in 2010. It's even less than $1,000 now, and it can take actually I think it's under a day or maybe around a day or two. It takes so in our first genome, it was 10 years. Now it's $1,000, and it takes usually a day or two to complete the sequencing. Now, your human genome is made of about 25,000 genes. When we first were looking at the number of genes in a genome, we actually thought for humans it would be more than like 100,000 because, or even a couple 100,000. But because we thought since we're such, we have these big brains and we're so advanced to everything else. And the reality is, again, we're just another species on Earth. We're not like unique. We're just part of the every other species. And it's just kind of our, I don't want to say arrogance, but like we just thought we were special. And even though we are kind of special, it's just not as we thought it was. And we actually have about the same number of genes as a chicken. I know it sounds weird, but yeah, we have about the same number of genes as a chicken. So we have, again, 25,000 genes around. Around 2% of your genome actually codes for proteins. The other 98%, they regulate gene expression. So we talked about this a little bit last chapter, but again, so only a small amount of your genome is protein producing. The rest just regulates that 2%. And again, it's not like having more genes or having more chromosomes is better. It's just how things are in life and how things have evolved. So you can see like the diploid number. Chickens have more chromosomes, the diploid number, but they have about the same. I think it's about the same number of genes. Yeah, you can see about the same number of genes. We have, again, we share close number of genes with like the other mammals because obviously we're mammals and we all evolved from a common recent ancestor. But then again, this is just how we evolve. So you can see it's just random kind of, but there is some evolution based kind of similarities. Okay. So that, again, that's its like own little mini section. New thing, genetic fingerprinting. So genetic fingerprinting is when we take like a sample of DNA and we're going to run it through a couple processes and create this genetic fingerprint for a person. This is almost like a fingerprint. Here, I can bring my video. It's almost like a fingerprint for your genome, and everyone has their own specific set of fingerprints in their genes, kind of. So they're not like actual fingerprints, but they're just these repeated sequences. And what we can run them through a machine called a gel electrophoresis machine. Now, these machines will separate DNA based on length, and everyone has these different lengths they have. I'm not going to get too bogged down in the details, but basically, the shorter strands of DNA go further in this plate, and the longer strands don't go as far. So what this plate does is it just kind of separates your DNA fragments into different lines, and we can use that those fragments to kind of identify like people at a crime scene or the paternity of somebody. So this was huge back in the 80s when they first figured out they could do this. You can see how like in a crime scene we can match the people. You can say, hey, this person was at the crime scene or not the crime scene. But we can also talk or see the paternity as you can see with the child here, which is the father here, and then the mom and dad, and try to figure out who came from the mom and dad. This is just a way that we can use your genome to figure out relation to a sample of DNA. All right, cloning. Again, this is totally just, it's not like a progression of genetic fingerprinting. It's just, hey, this is new and this is what we're dealing with. Replaces the nucleus of an egg with another or cell's nucleus. So again, you have an egg from, like I said, it's frog. We take out the nucleus. We take the nucleus from another frog, implant it into the egg, and that new organism will be a clone of frog B. It's just like an exact copy. And I love showing this video where you can actually get your dogs cloned for about $100,000 in South Korea now. So yeah, this is a new biotechnology. This was bigger back when I was in high school. Dolly the sheep, I think Dolly the sheep comes up next. Yeah, Dolly the sheep was the first vertebrate cloned, and we do cloning in other things, in other types of organisms. We've known it for years, but the first vertebrate that was vertebrate, I should say, that was cloned was Dolly the sheep. Now, again, totally different. This is something called CRISPR. This is something more recent. Come up, there we go. And what CRISPR is basically what was found again in 2012 in bacteria, but this allows scientists to edit genes. So what essentially this does is it's this protein we found in bacteria that if we give it a certain target sequence of DNA, it can find that DNA in a person and either cut it, edit it, remove it, do whatever. And this has huge implications. This was a Nobel Prize winning feature thing, discovery back in the early 2000s, I think it was, yeah, 2012 here, that really revolutionized biotechnology. You will hear a lot about CRISPR in the future, a lot, a lot, a lot. I'm not going to show you this video, but there's a lot of good information about CRISPR. All right, GMOs. GMO stands for genetically modified organisms. This is when scientists take a section of DNA from one organism and insert it into another organism, and it creates a beneficial trait. Usually, we do this with plants, but we do this with a lot of other organisms now. But do understand there's a lot of misinformation out there about GMOs. GMOs are a good thing overall. There can be negative side effects. I want to say not side effects, but kind of socio economic side effects, is that, if that makes sense. I'll give you for instance here of like a benefit: golden rice. It adds vitamin A to a like a rice or the rice, and you can see the vitamin A. There's a huge deficiency in vitamin A in these populations, and it really helps people in those locations because we're inserting a gene to make more vitamin A in the rice. BT corn. It kills certain types of like infestations and like insects, and it's because of this chemical that's produced that it's harmful to the insects, but not us. And you can see what that looks like between BT and normal corn. And again, we've been doing this for years. Making insulin. You can see what was needed back in the 50s to make 1 pound of insulin. Now we just use bacteria to make this insulin. Again, much more humane way of producing insulin. So yeah, there's a lot of benefits. Obviously, crop yield, development of better resistant crops, decreased use of pesticides, but there are also some negatives. The GM companies do regulate the prices of their GM crops, and they also may threaten non-threatening insects like the monarch butterfly. And monoculture farming can lead to problems in ecosystems as well. And there is the aspect of the economics of small versus large farmlands. And I'm not saying that's not a problem. That is definitely a problem. We have to work with these small farms and making sure that it's not just run by larger farms, and that these small farms can sustain the business of GMOs. But there's a lot of misinformation out there about. Let's see here. Like it's the GMO free fruit. Like just because it's a GMO doesn't mean it's bad. GMO products are beneficial in nature. So a lot of that there's a lot of misinformation or misguided thoughts about GMOs. All right, telomeres. These are the ends. Let me go back. These are the ends of the chromosomes, and what they do is they're again. This is this part of the non-coding part of the DNA, and this protects the innermost important portion of the chromosome. And what happens is, as the chromosome replicates, it's going to shave off the ends here a little bit by little bit. And what happens is, at the end, when you don't have any more, that's when cellular senescence occurs, which means the cell is going to die. And there are enzymes. Let me go back. There are enzymes that can help with replacing these telomeres at the ends. And this is something we didn't know about chromosomes, but you can see that this is going to be something that they've linked this with like aging, and like people with longer telomeres seem to live a little longer. So if there's a way that we could enhance these telomeres, there could be benefits of living longer. But again, there's a lot that's not known about telomeres, and we're still finding out. I give the analogy of the telomeres being like the aglets of a shoelace. Again, lots of good videos. All right, lastly, vaccines. I think this is the last part here. Vaccines. So this is a biological preparation that improves immunity of a particular disease. It stimulates the body's immune system to recognize foreign factors such as pathogens and viruses and bacteria. This is either typically a dead or weakened virus. And basically, I mean, everyone talks about the vaccine, the COVID vaccine. We're happy to have it now. A couple years ago when I was teaching this, I was like, we were talking about how there, just the development of vaccines and how difficult it can be, just because every virus is different, and the funding for these viruses, we need more funding. Obviously, past 2 years with COVID, there's been a huge push for these vaccines and a lot more support and a lot more resources provided for the development of a vaccine. So you can see each vaccine is going to be different because each virus is going to have these different. They're called the antigens on the virus. Our body produces new antigens for each virus. So whenever you see a new pathogen, like we saw, this graphic was from years ago. Obviously, it's exactly what we were talking about with COVID, but you don't have any antibodies for it. So your body then, or this vaccine, will produce your body to have these antibodies. So the new mRNA vaccine, it's a not to say new in the sense of new, like just like it was popped out of nowhere. We've been working with mRNA vaccines for more than 10 years now. But basically, what this does is this mRNA produces in your cells the spike proteins, and that's it. When your body recognizes these spike proteins, it will produce the antibodies for them. So it's like giving the instructions on how to produce the antibodies without actually giving the virus. It's a much easier and much more effective way of producing a vaccine. So the COVID-19 vaccine is an mRNA, which is a quote unquote newer type of vaccine, but we've been dealing with these mRNA vaccines for years and years and years now. Again, I show a bunch of videos when we talk about this vaccine. But the whole point, and like we've been talking about, it's I don't want to say it's very interesting that everything that I've been teaching about these vaccines from years ago, it played out. I remember with the end of the year, I would always show, and I will show, it's called Contagion. It's about a virus that takes over the world, and just how similar that was to what happened with COVID. I remember whenever like the COVID, starting 2 years ago, showing that movie years and years ago, and I said, my students, like, this could happen, this could happen, this could happen. I remember all my students, oh yeah, sure it can happen, and then it happened, and then I got all these this communication saying like, hey, I remember like watching that movie, and now it happened. So it's just interesting how that plays out. But basically, what we're trying to get to is something called herd immunity, where even if each person doesn't get the vaccine, or if people, because not everyone can get the vaccine. Obviously, with COVID, that like the younger populations, they can't. What we're trying to do is we're trying to get herd immunity. So even if people do still get infected, there's enough people who are immune to it, or who won't or just who will be immune either because of the vaccine or because they got it, so they won't it won't spread very very much like in these populations. You can this population where there's no immunity, it spreads very quickly. Some immunity, again, it still spreads. High amount of immunity, you can see it stops. It still might transmit, but it's not as much as without the herd immunity. I think that's it. Yeah. Okay, that's it.